You are a woman known to science only as SM, and for the first time in decades, a team of researchers is about to make you feel terror. You sit calmly in a medical chair, a plastic mask fitted over your mouth and nose. Through it you breathe normal air. Then, without warning, the mixture changes. A rush of 35% carbon dioxide floods your lungs. Your body seizes. A primal, suffocating fire ignites in your chest, and for the first time you can remember, you are overwhelmed by an unspeakable panic. You gasp, your eyes wide with a feeling you cannot name, and you scream for the doctors to stop. Here is how you arrived at this moment. Your story begins not in a laboratory, but in the quiet of an ordinary life. You grow up, you have children. In your 20s, you begin to notice small things. Your voice has always been a bit hoarse, a trait since childhood. But other changes are happening deep inside you, unseen and unfelt. You have an extremely rare genetic condition called Urbach-White disease. It causes tissue throughout your body to harden and calcify. For most with the disease, it manifests as skin lesions and a raspy voice. But in your brain, the condition is executing a stunningly precise attack. It is targeting two small almond-shaped structures on either side of your temporal lobes. These are your amygdala. They are the core of your brain's fear circuitry. As you move through your 20s and 30s, Urbach-White disease systematically destroys them. The tissue turns to useless calcium deposits. The change is not sudden. There is no single moment you notice fear is gone. It simply fades from your life like a color fading from a photograph, until the very concept becomes alien. Your unique condition eventually brings you to the neuroscience department at the University of Iowa. Researchers there, led by Dr. Ralph Adolphs and Dr. Daniel Tranel, have been searching for a way to understand the amygdala's role in human emotion. In you, they find a perfect, naturally occurring case study. You are open, friendly, and eager to help. You agree to a battery of tests designed to provoke a single emotion, fear. The researchers begin with movies. They show you clips from the most terrifying films ever made. Scenes from The Shining. The scariest moments of Silence of the Lambs. Jump scares from modern horror. You watch with detached interest. You find some scenes exciting, others simply boring. While other viewers would show a spike in heart rate and clammy palms, your physiological readings remain perfectly flat. You feel no dread, no anxiety, no terror. The scientists note your response: high levels of positive emotion, such as happiness and excitement. They show you pictures of gruesome injuries, natural disasters, and threatening faces. You describe them factually, but the intended emotional impact is completely absent. They ask you to fill out questionnaires measuring fear. You struggle to answer. How do you rate an emotion you no longer experience? It's like asking a person born blind to describe the color red. The researchers realize they need to move beyond images. They need to expose you to real, tangible threats. They take you to a local pet store that sells exotic animals. In a back room, they bring out trays of spiders and snakes, creatures that trigger a deep evolutionary fear in most humans. You are fascinated. You look at a large tarantula and a venomous-looking corn snake not with apprehension, but with intense curiosity. A researcher holds one of the snakes. You ask if you can touch it. You repeatedly try to touch it, telling the scientist how cool it is. You have to be physically stopped from getting too close. You later tell them you hate snakes and try to avoid them. Yet, in their presence, your body shows none of the physiological signs of fear or aversion. Your self-reported dislike is a purely intellectual fact, disconnected from any emotional or physical response. Your amygdala isn't there to translate the thought, snake, into the feeling, danger. The experiments escalate. The research team takes you to one of the most famously haunted places in America, the Waverly Hills Sanatorium in Kentucky. It's an abandoned hospital where paranormal investigation shows are filmed. For this test, the building is staffed with actors dressed as ghosts, monsters, and murderers, all prepared to leap from the shadows. You enter the sanatorium with a small group of control subjects. The others are tense, jumping at every creak and shadow. You, however, are buoyant and cheerful. You lead the group down the dark, decaying hallways. When a monster covered in fake blood leaps out and growls, the others scream. You laugh. You walk right up to the actor. You poke him in the chest, curious about his mask. The startled actor breaks character. Another actor dressed as a knife-wielding killer jumps from a hidden corner. You don't flinch. You smile and wave, telling him you're going to get him. You effectively scare the monsters. Over the entire hour-long experience, you register zero fear, only excitement. These controlled experiments are revealing, but it's your life outside the lab that shows the true consequence of your condition. The absence of fear is not a superpower. It is a profound vulnerability. Your amygdala is supposed to be an early warning system, a silent alarm that helps you detect and avoid danger. Yours is silent. You tell the researchers about an incident that happened years prior. You were walking through a park alone at night. A man approached you, grabbed you, and held a knife to your throat. He hissed at you, "I'm going to cut you." Most people would be paralyzed by terror. Their bodies would be flooded with adrenaline. Their minds would race. You felt nothing of the sort. You looked at him calmly. You later recalled feeling not fear, but a sense of divine protection. You told him, "If you're going to kill me, you're going to have to go through my God's angels first." The man was so taken aback by your utter lack of fear that he let you go. You simply turned and walked home. You never called the police. The event left no traumatic trace. On another occasion, your first husband beat you and held you to the floor, threatening your life. You fought back furiously, escaped, and left him. But you never felt afraid of him. You felt anger, you felt resolve, but the chilling dread that should accompany such a threat never registered. You have walked into dangerous situations, trusted untrustworthy people, and put yourself at risk time and again because the internal alarm that screams, "Danger," is gone. The research reveals another, more subtle deficit. You struggle to recognize fear in other people. When shown photographs of faces expressing different emotions, you can easily identify happiness, sadness, disgust, and anger. But when shown a face with wide eyes and an open-mouthed gasp, the universal signs of terror, you are stumped. You don't know what you're looking at. Using eye-tracking technology, the scientists discover why. When you look at a face, you, like most people, focus on the mouth and nose. But to identify fear, humans instinctively look at the eyes. The amygdala is what directs your gaze to the most salient emotional cues. Without it, your brain doesn't know to check the eyes for signs of danger. When the researchers specifically instruct you, look at the eyes, your ability to recognize fear dramatically improves. For years, the conclusion seems simple. The amygdala is the brain's fear center. Without it, you cannot experience or recognize fear from external threats. But a new scientist on the team, Dr. Justin Feinstein, has a different question. What about internal threats? What happens when the danger comes not from a snake or a man with a knife, but from inside your own body? This question leads you to the medical chair with the plastic mask. Dr. Feinstein designs an experiment using a medical-grade mixture of air with 35% carbon dioxide. For a healthy person, inhaling this gas is not dangerous, but it instantly tricks the brain into believing it is suffocating. It creates a powerful primal sense of air hunger that triggers an immediate and overwhelming panic attack. Dr. Feinstein wants to know if this internal signal of suffocation is processed by the amygdala, or if it takes a different route. He runs the test on you and on 2 other patients with the same rare bilateral amygdala damage. The result is shocking. The CO2-rich air hits your lungs. Instantly, your body overrides your conscious mind. Your heart rate skyrockets. You begin to sweat. You gasp for air, thrashing in the chair and tearing at the mask. For the first time since you were a young child before your amygdala was fully destroyed, you feel a raw, unfiltered wave of pure panic. You describe the feeling as a terror you have never experienced before. The experiment reveals something profound. The amygdala is essential for processing and learning from external threats in the environment. But the brain has another, more ancient survival circuit, likely in the brainstem, that responds to immediate physiological dangers like suffocation. Your brain can, in fact, produce a state of terror. It just needs a very specific kind of trigger. Your case, the decades of research you have participated in, has fundamentally rewritten our understanding of fear. Fear is not one thing, but many. There is the cognitive fear of a monster in a movie, the learned fear of a snake, and the primal bodily panic of not being able to breathe. Your amygdala's absence means you cannot form fear memories. You cannot develop post-traumatic stress disorder. But it also means you cannot learn from mistakes that should have terrified you. The protective instinct that guides billions of people away from dark alleys and dangerous situations is not there to guide you. Fear, your life demonstrates, is a critical tool for survival, a gift from our evolutionary past. That's the story of SM, the woman who taught the world what it means to be fearless. After the carbon dioxide experiment, SM had no memory of the panic she felt. Her damaged brain was unable to form a new fear memory, and she stated she would gladly do the experiment again, unafraid. Subscribe to Enlighten Me for more.